So Alain also carries out other studies. One of them is studying the impact of the different collection intensities and methods. So he put some plots and looked at what happens to some plants if you collect none of the plants, or 20%, 50%, or even 80% of the population. And of course, it has a major impact. So that helps to guide of how to manage the resource. And of course, he's also doing some studies on different stages of the plant life cycle. And this is done in close collaboration again with the elders, because they have also traditional knowledge concerning this. The other thing that the elders have advised us is that there was differences between the plants growing either between the southern communities and the northern communities. And if you remember, there's also some communities on the coast of James Bay and Hudson's Bay, even the highest one, whereas there are some inland communities. So we looked at also geographical. I won't give you any of the results for that for lack of time, but this is the types of things that we are doing. John Arneson here at the University of Ottawa, as I said, is responsible for the chemistry part of the components of the plants. So he's prepared the plant extracts, and he took two approaches. One is a more classical phytochemistry approach of doing 80% alcohol extract of the material. It's a stronger solvent than water, so it extracts better some of the components. But he also used hot water extracts. But the way that the more classical from the ethnobotany side, the phytochemistry side, is like a tea. So it was 15 minutes at 90 degrees Celsius. And he adapted his methodology to reflect more what the elders were doing, which was going much more like 2 or 3 hours at near boiling. But it was Thor has been working with medicinal plants for 30 years. But most of the time he was, like as he is now, he's in Costa Rica, the little bugger. So he's been studying plants of the Amazon and things like that. And when he joined the project, when we got together, there was very little known about these boreal forest plants. So there was very little methods, no reference standards. Those are known chemicals that you can synthesize or extract, purify, that serve as standards. Often many of the markers were unknown, and obviously in terms of activity, active principles, very few were, or essentially none. Also, his work is to isolate and identify the active compounds. So he uses classical analytical chemistry tools, like for identification, it's like infrared and nuclear magnetic resonance or mass spectroscopy, but also just from chromatography. And he has a library of profiles. So this is called deconvolution. It's identifying known compounds. So he's very good at, and mostly for phenolics. Just to give you an idea, this is the screening of the first 8 plants, the same that I showed you the pictures. And you know, these are very common. For those who know phytochemistry, these are quite common in many plants, and it was not surprising to find them. And they have known biological activities as well. Now let's get into the crux of this pharmacology and toxicology. And I will show you 3. So we first do it in a screening with test tubes and cells. I will show you this in a second. Then we use animal models, and I will show you some mechanisms of action also. So most of this is done for the what we call primary anti-diabetic activity. This relates to cells involved in the response to insulin. So you'll recognize the liver, the skeletal muscle, and the fat that I mentioned earlier in the talk. We also added the intestines because glucose, that's where it's absorbed. So if you can reduce the absorption of glucose, that would be a good way to reduce blood sugar as well. Mark Prentke, who is the head of the Montreal Diabetes Research Center, is a collaborator, and he's an expert in the pancreatic beta cells which produce insulin. So we also have, so these are all cells that we grow in Petri dishes, and we can put the plant extracts and check the activities for all of these things that I explained to you. And Lina Musalam is a very important person. She's the lab coordinator. She coordinates things in my own lab, but also with all of these labs. There are 7 labs in 3 universities, so it's a quite, it needs a lot of coordination. So those are the primary anti-diabetic activities, and we can use these afterwards to try to identify active principles by what's called bioassay-guided fractionation, and I'll give you an example later on in the talk. Again, I just put this back to tell you that once we see activity, then we can go a bit deeper, try to identify where and how the plants are acting by looking at all these pathways that I don't want you to remember. Okay. Then there are another set of studies still what we call in vitro, so in test tubes or in Petri dishes. And these are so-called secondary anti-diabetic activities. So those are related more to the complications of diabetes. So Stephanie Bennett looks at what's the equivalent of diabetic neuropathy, so the pins and needles that you have. So she's looking at the protection of neurons in culture against either too much glucose or too little glucose by the plants. Thor is involved in the looking at whether the plants have any effect on inflammation. And also Tim Johns at McGill looks at antioxidant and anti-glycation. When you have a lot of sugar in the blood, as diabetics do, sugar tends to stick onto proteins. And actually, hemoglobin A1C is like the gold standard. This gives you an idea of how your glucose has been doing over about a three-month period, which is the life cycle of a red blood cell. Okay, so we can look at how plants can protect against glucose sticking onto proteins in test tubes. This is what we've been doing. And then Brian, who's sitting in there with us, is an expert in toxicology, and especially for what is a major concern is the potential interaction between the medicinal plants and the medication, the diabetes medications. So there are enzymes in the liver mostly, and also in the intestine that metabolize drugs and that can be interfered with. So he's studying that. There's transport or efflux of your body tries to get rid of the drugs, so that happens also in the intestine. And he's been doing some work also on cardiomyocyte toxicity. So these are the what we do in test tubes or in Petri dishes. We also do work with whole animals. And the model we've had most success with is called the diet-induced obesity model. So we take these little C57 black 6 mice that you see on top here. If we just feed them a normal chow diet, which is based on 4% of the energy coming from lipids, that's what they look like. If you put them on a high-fat diet where 35% of the energy is derived from fats, after a couple of weeks, that's what they look like. That's the same age, the 2 mice here. So we have 2 ways of studying the plants. If we want to see if the plants can prevent obesity, then when we start, so we always wait till the animals are 4 weeks of age, and then we start them and we treat them for 8 weeks to get to, after 8 weeks, they get to a sort of early diabetes. They start having a higher blood sugar and higher sugar in their blood, etc. Or if we want to really have them go to a more serious stage of diabetes, we prolong this to another 8 weeks, to a total of 12. So when we want to look at prevention, we just start the plants at the same time as the high-fat diet and compare it with the animals not receiving the plants or to the normal animals, healthy and lean animals. When we want to look at the effect on treatment, so we let the mice get diabetic. So by 12 weeks, then we start. So we have the fat diet for 8 weeks, and then another 8 weeks with the plants. And I'll show you some examples of these results later on. I can't go into the detail of all the data that we've done, so I summarize this because we have almost 20 papers published in the last 5 years with all these 7 labs working together. We've been quite productive. But I'll give you some key points. Of course, as I showed you, we developed a novel ethnobotanical approach that was validated. We screened these 17 plants. We found none that had any effect on the production of insulin by the pancreatic cells in the Petri dish. We found 10 that could increase the amount of glucose going into either the muscle or the fat. 8 that could enhance the development of the fat cells, and I will show you some examples later on. Some that were 10 that were protecting neurons, as I mentioned, and other also in endothelial cells. These are the cells lining your blood vessels, so it's related to the cardiovascular atherosclerosis and things like that. 9 of the 17 plants are some of them as potent as vitamin C in terms of antioxidants. We found almost half of them that could inhibit the transport of glucose in the intestine, so reduce the amount you actually take in. This is another way. Some had or anti-inflammatory activity, and this was related a lot if it was the water extract or the ethanol extract. I can go in greater details if you want in the question period. We found 12 that could prevent this is advanced glycation end products, this AGE, which is the glucose sticking to proteins. In terms of their potential interaction with drugs, well, luckily most of them had low to moderate, but a few could inhibit some of these drug metabolizing enzymes. So this raises little red flags. Brian is always, and it's very important that he raises these little red flags. So we're paying a lot of attention to that. Sorry. Out of these 17, as I told you from the work plan initially, a few of them we have tested in the animal models. So we have confirmed for 6 of them that they can reduce blood sugar in these fat pre-diabetic animals. And I'll give you the story of one plant where we found some anti-obesity activity, which is very interesting. We've identified several novel compounds and found the mechanisms of action. I'll give you some details about this in a second. So I just wanted to give you some examples of these tests so that you get a grasp of this. And I'm taking only data that hasn't been published. So what we will be looking at is plants that can reduce the production of glucose by the liver. Okay, we haven't, as you showed in the summary, we hadn't looked at that yet. So Abir Nashar, a graduate student in my laboratory, is looking at that. There are 2 enzymes. One is glucose 6-phosphatase. That's the last enzyme that cleaves this phosphate to enable the release of glucose by the liver. Okay, and what you're looking to do is decrease its activity because that will reduce the amount of glucose that can get out of the liver. As I told you, the liver is also a major storage site. So when there's too much sugar, it stores it into this glycogen, animal starch, if you want. So we're looking at the activity of glycogen synthase, which is the enzyme that makes this polymer of glucose. What you have here on the right is the activity of the inhibition of the activity of this enzyme glucose 6-phosphatase, and you see insulin reduces it by about 60%. And you see that we have a few plants that are just as powerful as insulin. And again, these are not published, so you don't have the names. You just have codes for the plants. So 9 of them had a significant effect for decreasing this glucose 6-phosphatase, and 10 of them had significant effects. And some of them are incredible. This is the effect of insulin here. So it's still a doubling or more than 2 times the effect without insulin. But look at this. These 2 plants are amazing. They are very, very powerful. And one of the plants that we're paying most attention to is one that combines these 2 activities. So AD02 here is a very strong inhibitor of glucose 6-phosphatase. And look at this. It's the most potent stimulator of glycogen synthase. So if you have both happening, then that should really reduce the amount of glucose being put out by the liver. Of course, this is in Petri dishes, so we have to move on now to animal models. Then for the mechanisms of action, and don't get scared. I know it's, I'll make it easy and simple for you. This is just the different components inside the cell that respond to when insulin touches its receptor. This is the insulin receptor. You have many components activated in the cell. We're focusing on one of them called AKT here. That eventually leads to the movement. The person who helps us with making this into lay language calls these are glucose transporters, and she calls them dump trucks. So you move the dump trucks to the membrane to be able, they can take the sugar from the outside and bring it into the cells. Okay? And what insulin does is increase the number of dump trucks that go to the membrane. You can also have that by, and this is in the muscle, as I told you, by energy depletion. If you work out, then your muscles, they eat up the ATP, which is your chemical energy. And this enzyme, AMP kinase, responds to this change here of energy and can stimulate the same, putting out the dump trucks on the membrane. And this can happen also either by the exercise, as I told you, or by problems at the level of the mitochondria. These are your little powerhouses inside your cells that produce the ATP. And so these 4 boxes are the sites that we looked at to understand how the plants were working. And I'll walk you through this really quickly.